Yes, thank you very much. So we move on to our second plenary speaker for today's session. And our second speaker is Jef Huisman. Jef has made a very significant contributions in a truly remarkable range of disciplines, including aquatic microbial ecology, biological oceanography, theoretical ecology, ecophysiology, and photosynthesis research. His research spans fresh, brackish, and marine waters, and I have to say that he's truly an integrative scientist in every sense of the word. Jef carried out both his undergraduate and doctoral studies at the University of Groningen in the Netherlands, and after 2 postdocs, he started as a faculty as a professor in aquatic microbial ecology at the University of Amsterdam in 1999, where he has been ever since. Since 2017, he has also been the head of the Department of Freshwater and Marine Ecology of the University of Amsterdam. His research combines mathematical models, laboratory experiments, and field studies to investigate species interactions in aquatic ecosystems. He has worked on a wide variety of organisms ranging from microbes to whales, so it doesn't get much wider than that, except if you add viruses, I guess. But much of his research has focused on plankton communities. He has made key contributions to resource competition theory, advancing models and experiments to assess how nutrients and light affect the species composition of freshwater and marine phytoplankton communities. Among many other issues that they are investigating, his team investigates impacts of climate change on plankton performance and distribution, and in particular, the potential effects of rising CO2 and temperature. And in this regard, his group has made also key contributions to our understanding of the origin, dynamics, and regulation of harmful algal blooms, focusing mainly on cyanobacteria, but not only, and to our capacity to predict these phenomena under scenarios of environmental and climatic change. So Jef, his team, and collaborators have more recently extended these lines of research to encompass cutting-edge remote sensing applications of water properties that allow unprecedented insight into the function and distribution and the biogeography of photosynthetic organisms in the world's surface water. So in its plenary, he will be sharing with us some of his insight, and I really look forward to that. So I give you the floor, Jef. Thank you very much.
Thank you, Paul, for this very kind, very nice introduction. I really appreciate it. And I'm still digesting the stunning pictures of Bernard Lehner. Really, really beautiful. Now I'm trying to upload my talk. And I also have to start with an apology. I'm not a remote sensing specialist. Maybe that was already clear by the introduction that Paul gave. I'm a more plankton ecologist that is using remote sensing to understand how plankton behave and are distributed over our planet. And that's how I wanted to use also in this presentation. So I want to take you on a tour. We go from the molecular vibrations of the water molecule to try and predict the global distribution of photosynthetic pigments across the lakes and oceans of our planet. But before doing so, I first want to congratulate Elena. Elena, congratulations with the Hutchinson Medal. And I think your talk and your approach can also be very inspiring for remote sensing specialists, because your approach is to use traits. And I think one of the major challenges for remote sensing is to also try and to detect and see those traits to make predictions of how species are distributed and how they will respond to environmental change. So here we see one of the great results that have been obtained by ocean remote sensing. So it has given us a view of the global distribution of phytoplankton across the world, in this case shown by the chlorophyll concentrations in the oceans and on the terrestrial world. And we can also use it to see how this varies over time and space. Now, there are many challenges in remote sensing, and Bernard knows much more about this than I do, and he already explained a lot about it. But one of the challenges is to get a better and better spectral resolution, so that we can see more of the underwater light color and maybe use that to distinguish different phytoplankton traits. And one of the key traits that I'm talking about here are the photosynthetic pigments of phytoplankton. And mostly remote sensing, not only, but a lot of the remote sensing has focused on chlorophyll. But chlorophyll is not the only pigment of phytoplankton, as shown here in these pictures. So you see a colorful selection of cyanobacteria that were all isolated from the Baltic Sea. And the colors vary from bright green to pink to dark brown. And we also know from our taxonomy textbooks that we have a wide diversity of different phytoplankton species, and their taxonomy is actually to a large extent based on their pigmentation. So we have red algae, green algae, brown algae, golden algae, and so on. So why do we have this tremendous diversity of colors? And what are these species using these colors for? Well, one hypothesis might be that actually light is not a single resource for phytoplankton, but it's a whole spectrum of resources. It's a full light spectrum, and different species might use different chunks of this light spectrum, as shown in this graph here. So you see red cyanobacteria, they're very effective in absorbing green light. And you see, for instance, green cyanobacteria, they're very effective in absorbing red light. So one of my former PhD students, who later became also a postdoc in the lab of Elena Lietzman, is Maaike Stomp. And she did a very elegant experiment with two cyanobacteria, a green cyanobacterium and a red cyanobacterium. And she put them together in competition. And what you really need to realize is that green cyanobacterium looks green because it is not good in absorbing green light. It reflects the green light. That's why we see it as a green cyanobacterium. But it is very good in absorbing other colors, for instance, in absorbing red light. So if you put these two species in competition in red light, then Maaike showed that the green species wins. But if you put the two species in competition in green light, then the red species wins. So you have always to do this complementary thinking. Think in terms of complementary colors. So different colors select for different phytoplankton species with different photosynthetic pigments. So that raises the question, which kind of spectral environments, light environments, can we find in aquatic ecosystems? And is it just a free distribution of different colors from the violet blue in the clearest oceans to the darkest brown in very turbid lakes? Or are there some environmental constraints on the light colors that we can find in aquatic ecosystems? And actually, Maaike showed a few years after those experiments, he showed in a very influential and very intriguing paper that actually vibrations of the water molecule play a very important role in shaping the underwater light spectra. Unfortunately, Maaike Stomp and also another colleague of mine, Hans Matthijs, they passed away a few years ago, so they cannot tell their story anymore. But luckily, both Lucas Tell and I can tell the major insights of this work. So let me try to explain you what these molecular vibrations of water, what they do to the underwater light environment. So the first thing we start with is the vibrational modes of the water molecule, and there are three fundamental vibrational modes. One of them is the symmetric stretching. So you have the two hydrogen atoms and the single oxygen atoms, and both hydrogen atoms go back and forth in symmetry. That's symmetric stretching. If you were joining the yoga lesson at the ASLO meetings this morning, you can also help me and really make this symmetric move, right? So the other motion that they can do, that's the bending. So you can see it's a different motion, and it goes up and down. The screen is too small to show to you, but you can see the bending. It's also a fundamental vibrational mode of water molecules. And the third vibrational mode, that's the asymmetric stretching. So here, when one hydrogen atom is closer to the oxygen atom, the other moves further away. So it goes in anti-phase, these two hydrogen atoms. So these are the three fundamental vibrational modes of water. And when you do this exercise, you feel that this motion requires some energy. And they can obtain this energy by absorbing photons, so by absorbing light. Water molecules absorb light, then they can do these vibrations. And we can see that in the absorption spectrum of pure water. So that's on this graph here. It's the absorption spectrum of water from the far into the UV at 100 nanometers to far into the infrared at 10,000 nanometers. And the fundamental frequency for the bending vibrations occurs here at this V2, at this wavelength of about 7,000 nanometers. And then the symmetric stretching requires a little bit more energy, so it occurs at a shorter wavelength of about 3,000 nanometers. And the asymmetric stretching has a similar energy requirement, approximately. So it also occurs here at about the 3,000 nanometers. And that's why we see this very large peak in the absorption spectrum of pure water. So in a sense, water likes to absorb these wavelengths because it then can make these vibrations, the symmetric and asymmetric stretching. Now it can also be that water absorbs a photon with a higher energy content that just matches two vibrations together. So here at this wavelength of about 2,000 nanometers, one photon contains the amount of energy that's required for one bending vibration and one asymmetric stretching vibration. So you can interpret it as a kind of second harmonics. Then here is the second harmonics of the two stretching vibrations at an even shorter wavelength. And then there's another peak in the absorption spectrum of pure water. There's another peak over here that corresponds with the so-called third harmonic. So a photon at this wavelength provides the energy for all three vibrational modes of the water molecule. And now we move closer and closer to the visible spectrum, and you can see even more peaks and valleys in this absorption spectrum of pure water. So let's zoom in to the visible range. We're now looking at a graph that shows the absorption spectrum of pure water of H2O between 400 and 700 nanometers. And again, we see these tiny shoulders in the absorption spectrum. And these shoulders, they correspond to the different harmonics of the vibrational modes. So we have the 8 harmonics, where one photon provides the energy for 8 different motions of the water molecule. We have the 7th harmonics with a tiny peak, there's a tiny shoulder here at the 6th harmonics, the 5th harmonics, and something that's called the 4.1 subharmonic. So we see these different vibrations back in the visible range. Now, the water molecule itself is, of course, not the only substance in water that absorbs light. There are also very important other substances, and one of them is colored dissolved organic matter, or CDOM. It's also absorbing light, and here you see the absorption spectrum of CDOM, the typical absorption spectrum. It absorbs a lot of light in the blue and the violet part of the spectrum, and it absorbs very little light in the orange and red part of the spectrum. So that's why water with a high CDOM content often looks like brownish red, because that's the color that is not absorbed. And if we have waters with different CDOM concentrations, as here shown in these 3 LMIs, then it will change the height of this brown curve, right? So if you have more CDOM, then this brown curve will be lifted. And if you have water with very little CDOM, then this brown curve will be very low. But it's always having the highest absorption in the blue and violet part of the spectrum. With one of my PhD students, Tatio Holtrop, we simulated the underwater light spectrum for 50 different waters, and each water had a different CDOM concentration. So here you see the absorption spectra of these 50 different aquatic ecosystems, going from the blue ocean, where the absorption spectrum of the blue ocean is almost similar to the absorption spectrum of pure water. And then we add more and more CDOM, more and more organic matter to the water, and what happens is you get more and more absorption here in the violet and blue part of the spectrum until we are up here in a very humid lake with an extremely high CDOM concentration. So from these 50 absorption spectra, we calculated the underwater light spectrum at the euphotic depth. And the euphotic depth is the depth where you find 1% of the surface irradiance. For instance, if you take the blue line over here, the absorption spectrum of the blue ocean, then this type of water creates an underwater light spectrum at the euphotic depth, and that's quite deep in the open ocean at about 100 or 150 meters depth. You find this underwater light spectrum. It's dominated by violet and blue colors. Now, if you take the yellow spectrum over here, it's typical of a mesotrophic lake. Many of the lakes that were shown in the previous lecture have absorption spectra that look a bit like this yellow curve, and then you get an underwater light spectrum that's dominated by the green wavelengths. Now, if you go to this very humid lake, this pink line over here, you get an absorbed, very turbid, so the euphoric depth is very shallow, only at about 50 centimeters perhaps, but it's dominated by light in the orange and red part of the spectrum. So we have this landscape here. This is an overlay of 50 of these underwater light spectra, and together these 50 underwater light spectra form a landscape of peaks and valleys. And what is most striking is that these valleys that we see in this landscape, they are created by the harmonics of the vibrations of the water molecules. So these tiny shoulders over here, they cause deep gaps in the underwater light spectrum. And the reason that these tiny shoulders can cause deep gaps is because of the exponential nature of light attenuation. As you know from Lambert-Beer's law, light declines exponentially with depth. This also means that a tiny bit of extra absorption at this shoulder is magnified exponentially, so it creates a deep gap in the underwater light spectrum. So what does this tell us about photosynthesis? Well, it creates a kind of landscape, an energy landscape, to which photosynthetic organisms can tune their photosynthetic pigments. So basically, we can distinguish 5 different spectral niches here. The violet niche between the 8th and the 7th harmonics, the blue niche between the 7th and the 6th harmonics, the green niche between the 6th and the 5th harmonics, the orange niche between the 5th and the 4.1 subharmonic, and the red niche from 4.1 to 700 nanometers. And basically, our expectation is that optimal photosynthesis is achieved if you can tune your photosynthetic pigment to these underwater light environments, so to the green niche or to the blue niche, to the violet niche, and so on. So let's look how these spectral niches match with the absorption spectra of different photosynthetic organisms. We'll first have a look at Prochlorococcus. And Prochlorococcus, you might know, it's actually the most abundant photosynthetic organism on our planet. It's widespread in the oligotrophic ocean. And there are different types. There's a low light adapted ecotype and a high light adapted ecotype. They're all characterized by the photosynthetic pigments diphenylchlorophyll A and diphenyl chlorophyll B. And in that sense, they resemble, in a way, green algae and terrestrial plants, which have chlorophyll A and chlorophyll B. Now, these are the absorption spectra. One is of a highlight adapted Prochlorococcus, and the other one with more diphenyl chlorophyll B is of low light adapted Prochlorococcus. And what you can really nicely see is that these are well adapted to the violet and blue niche that we find in the oligotrophic open ocean. Now, if we look at other cyanobacteria have another different pigment composition, also different structural organization of the pigments. So many cyanobacteria have a so-called Phycobilizone. The Phycobilizone is a large light harvesting antenna that is mostly, not exclusively, but mostly on top of photosystem 2. So they can absorb light by these light harvesting antenna and then transfer the light energy to the reaction centers of their photosystems. And the pigments in these phycobilizomes are so-called phycobili pigments. And in essence, there are 3 different types of phycobili pigments. One is called phycocyanobulin, or PCB. Another one is called phycoerythrobilin, or PEP. And the third one is called phycourobulin, or PUP. So in my talk, we'll talk about PCB, PEP, and PUP. And these 3 different pigments can be contained in different amounts in the cells of cyanobacteria, in the phycobilizomes of cyanobacteria. So some species, they have mostly PCB as their phycobili pigment, and this is a typical color of such a cyanobacteria. Other cyanobacteria contain much more PEP in their phycobilizome, and then they can look pinkish. And other cyanobacteria can contain much more PUP, and then they have this more orange or red-like appearance. Now here I show you the absorption spectra of 8 different strains of cyanobacteria. They're all Photosynthesis strains with different contents of PUP, PEP, and PCB. And do you see the beauty of this graph? Because what the real beauty is of this graph is that each of these pigments captures a different spectral niche. Right? So they contain chlorophyll A in the reaction centers, and chlorophyll A absorbs in the violet part of the spectrum, in the violet niche, and in the red niche. It has 2 absorption peaks. And the PUP absorbs mostly in the blue spectral niche, PEP absorbs mostly in the green spectral niche, and PCB absorbs mostly in the orange niche. And the absorption peaks are very nicely separated. So these valleys here, they really are at the 6th harmonics, and here at the 7th harmonics, and here at the 5th harmonics. So, and here at the 4.1 subharmonics. So each pigment captures a different spectral niche. They're well tuned to the light color that's dominating the different aquatic ecosystems. So how can we use this with satellite, combine this with satellite remote sensing? Well, what we can do is we can look at the emergent color, the emergent light flux that's captured by satellites, and combine this emergent light flux with the radiative transfer model to predict the underwater light color from remote sensing data. So if we know the underwater light color, then we can also tell like which spectral niches are dominating in these aquatic ecosystems. I show this here for one part of the world. This is the east coast of the US. You may recognize Chesapeake Bay, that's this bay over here, and here in the south is Pamlico Sound, here's the Atlantic Ocean. And what we can see is that the green spectral niche dominates mostly in coastal waters. That's where you find a lot of green underwater light colors. If you go further into the ocean, it becomes more the blue part, you get the blue niche. And the orange niche, it dominates mostly in the upper part of Chesapeake Bay and also here in the lagoon section of Pamlico Sound. So more in the inland waters. And this is for April 2016. You can also imagine that these spectral niches vary during the seasons and over time. So let's look at the more global level. Here in the top graph, you see a map with the global distribution of the violet niche, also in April 2016, obtained by satellite data. And what you can see is that the violet niche mostly dominates in the subtropical gyres, so the very clear blue ocean waters. And we just saw that the diphenyl chlorophylls of Prochlorococcus indeed capture the violet niche very well. And indeed, there's a very nice match between the distribution of the violet niche and the global distribution of Prochlorococcus, in this case obtained from a paper by Flombaum in PNAS 2013. So they match very well. Now if we go to the blue spectral niche, again here we have a prediction of the global distribution of the blue spectral niche in April 2016. And you can see it dominates mostly in the temperate open ocean waters and also in the equatorial upwelling regions. And in these regions, we find often cyanobacteria that are very rich in this phycobilizone pigment, PUP. And PUP, that's specifically this pigment that is absorbing in the blue part of the light spectrum. And we confirmed this with data from Tara Oceans expedition, where they mapped the occurrence of the PUP-rich cyanobacteria. So they are mostly found in these temperate oceanic waters. Now, if you look at the green spectral niche, the green spectral niche is mostly confined to coastal waters or to inland waters, and also in the Baltic Sea here, in the Caspian Sea, the Chinese Sea. We already saw the Chesapeake Bay areas, upwelling areas over here. And in these areas, we often find the PEP-rich cyanobacteria, which also makes sense because that's where they have their absorption peak. They have their absorption peak here in the green spectral niche. Then if we move inland, then in inland waters, we often find cyanobacteria that contain either high contents of PEP or high contents of PCB, phycocyanobacterial. For instance, the Microcystis, a toxic bloom-forming cyanobacteria, that's a cyanobacterium that's rich in PCB, has a high content of phycocyanobacteria. So you can see this here in this map of the Great Lakes area. Then Francis Pick sampled those lakes and looked at the relative abundance of cyanobacteria with PCB and cyanobacteria with PEP pigments. And you can see these are the dominant ones. And the PEP-rich cyanobacteria are mostly found in the more mesotrophic lakes, and the PCB-rich cyanobacteria often bloom in the eutrophic lakes, where the orange niche is the most important. The orange is the most important underwater light color. So with this presentation, I hope to have shown you that the vibrations of the water molecule create deep valleys in the underwater light spectrum. And in this way, they create 5 spectral niches in the PAR range. And the photosynthetic pigments on our planet are tuned to these spectral niches. So different phycobilizone pigments, which we find in cyanobacteria, but also in red algae and in cryptophytes, they capture the different spectral niches like the orange or the red or the green niche. And these spectral niches can be detected and mapped by satellite remote sensing. So we may use satellite remote sensing to characterize the underwater light colors and use that information to predict what kind of taxonomic groups of phytoplankton we may expect in these different waters. And with that, I come to an end of my talk, and I want to thank all the collaborators who worked with me on this exciting topic over the several years. Thank you.